streaming. I'll wait 15 seconds and start talking. All right. Well, welcome everyone to our Ratios Matter seminar series. And for this installment, it's a little bit different than the last. So we have 3 speakers this time. And I'm really excited because we have 3 really awesome young up-and-coming stoichiometrists who are going to talk about their work today. So first off, we're going to start with Annie Laylock, and she is currently at the University of Alabama, sort of has a superstar team of advisors that do a lot of stoichiometry. And she's going to be talking about some work that she's doing, I think based up in Alaska, looking at the effects of ecological stoichiometry constraints and hydrological regime on functional trait diversity of aquatic insect communities.
Great, thank you so much. I'm very excited to be here. I hope everybody can hear me. I get a thumbs up. Yeah, so I am actually going to be talking about work that I did back at my undergraduate institution, Middlebury College, with Eric Moody. I'm working on a different project now. Obviously, it has to do with stoichiometry because everything does. But this project was my undergraduate thesis, the effect of ecological stoichiometric constraints and hydrologic regime on functional trait diversity of aquatic insect communities. So just getting started. Oops, no. All right. So we know that aquatic invertebrate communities experience stoichiometric shifts due to natural seasonal oscillations, including variation in drought and flood, timing and duration, which change environmental factors like average temperature, precipitation, light availability, and many other things in the environment. So this variation in basal resource stoichiometry and flow regime, they alter community composition, including competitive dynamics, which drive functional trait diversity of these aquatic communities. So functional diversity, as we know, or trait-based analyses help ecologists study and understand and explain the species distribution and community composition across environmental and disturbance gradients through the use of trait data to predict both individual as well as assemblage responses to environmental disturbance events or just seasonal changes. So we know a more functionally diverse community as well as a more biodiverse community is more resilient to environmental disturbance. So this is important in the larger conversation of human impact. So we know that anthropogenic impacts disrupt these competitive dynamics, which, as I said, were formerly constructed by seasonal oscillations in nutrient availability and flow predictability through land cover change, nutrient additions, et cetera. So it's important in this larger conversation of human impact and climate change driving environmental impacts or environmental change. So there's evidence that both hydrology and stoichiometry could be major drivers of functional diversity at regional scales, but we have yet to explore this at a continental scale. So this project sought to ask the question, are ecological stoichiometric constraints the dominant drivers of benthic macroinvertebrate functional trait diversity when compared to other constraining environmental factors like flow regime at a larger, broader spatial temporal scale? So our hypothesis was that functional diversity of benthic macroinvertebrate communities will decrease with increased hydrologic variation and algal P as taxa exhibiting functional trait states favored by limited phosphorus availability dominate aquatic communities. So how did we do this? In order to analyze this relationship between the functional trait diversity of aquatic insect communities in the United States and their chemical environments across a large spatial temporal scale, we use data from the National Ecological Observatory Network, or NEON. From the 34 aquatic sites that NEON samples 3 times per year, we use the aquatic insect and periphyton data from the 19 low-dick freshwater sites, including in Puerto Rico and Alaska, over a 4-year period. So that was all the data that was available. When I began this project, there was only 2 years of data available, but since I've graduated and more research students have worked with Eric over at Middlebury, they've expanded the database, which is really exciting. So with this NEON data, we created a database of functional trait data and relative taxa abundances for all taxa observed within all of our NEON data that we collect or that we compiled. So we chose 4 functional traits, which we decided were 4 functional traits incredibly impacted by phosphorus availability specifically. This included volcanism, development rate and seasonality, maximum body size, and trophic habit. We used 2 metrics to measure functional diversity. These were functional richness, which you can think of as similar to species richness, so the range of trait space occupied by each community, and functional dispersion, or the relative abundance of traits in each community. So additionally, we measured total invertebrate abundance, and we included that in our analyses. So our main objective, including hydrologic variation, was to quantify magnitude, duration, and frequency of flow events to study the impact of flashiness and predictability specifically on functional trait diversity, because we know that flashy unpredictability or predictability have huge implications on aquatic invert life history adaptations. So we wanted to look at both long-term and short-term variability and predictability. So we looked at long-term predictability, or really how predictable the timing and magnitude of floods and droughts are in the study area. We characterized this using high flow variation, or variance in the most extreme flood each year over a period of 30 years. And that's represented in the figure on the left. And we also looked at short-term variability. And we wanted to know, how frequent floods and droughts were during the period of which we use, from which our NEON data was from. Sorry, let me say that again. How frequent floods and droughts were during the period for which we have our NEON data, so over the 4 years. And this was characterized using skewness. And skewness was defined as the probability density function of daily mean flow over 1 or a few years. And that's represented as an example on the figure on the right. So basically just a shorter term look at variability in flow. So diving into our results, this is obviously a small selection of what we did, and there's continued greater analysis happening over at the Moody Lab at Middlebury. But starting with just to orient us, so we have functional richness as our y-axis, functional dispersion, and macroinvertebrate density or abundance. And this is looking at the relative impact of algal C2P. So we found that increased algal C2P or decreased phosphorus availability decreases total abundance, which is supported by our, supports our hypothesis, but it only had a weak negative effect on functional diversity. So no significant impact on these metrics of functional diversity, richness, and dispersion, which is interesting. So moving on, we found unpredictable flow regimes or increased unpredictability, reduced functional dispersion and functional richness. Again, sorry, we're looking at functional richness, dispersion, and the macroinvertebrate density, and their impacts, the impact of high flow variation on all of those variables. So this unpredictable flow regimes reducing functional diversity, that supports our hypothesis, and leading likely to trait convergence. So seeing that the number of traits and trait combinations reduced in a disturbed community, and likely converging on particular traits that are selected for this environment. And finally, looking at that short-term variation. So we saw that short-term hydrologic variation or increased skewness, more highly skewed streams, decreased functional diversity as well. So a decrease in functional richness and dispersion, again, suggesting this convergence on particular functional trait states that are selected for this more selective environment. So diving into these results, what we could learn from the hydrology perspective, we found that unpredictable high flow events and more highly skewed streams appear to drive convergence on trait states like univolcanism, fast development, and small body size at this continental scale that we were looking at, which makes sense that this more selective, more stressful environment is selecting on these life history states that are more resistant or resilient. And then from our analysis of the impacts of stoichiometric constraints, we found that decreased algal C2P was a less significant driver of functional diversity at a continental scale. But lower algal C2P, greater phosphorus availability is associated with higher benthic macroinvertebrate densities, which makes sense from a stoichiometry perspective. So just some final thoughts, looking at the broader picture. Flow regime and algal C2P or phosphorus availability both constrain insect life histories and thus are drivers of functional trait distribution, but we see they differentially impact that community composition. So during this research, as I was writing my undergraduate thesis, I read a lot about the impact of humans changing flow regimes through land change and especially land cover change. These changes in flow completely change community makeup. And this will continue to disadvantage native flow adapted, nutrient adapted insects, making space or making competitive space for non-native invasive generalists likely. And this reduced functional and taxonomic diversity of aquatic communities make ecosystems more vulnerable and less resilient, as we know, to dynamic environmental disturbance, which, as we know, will continue to increase in intensity and frequency with climate change, with continued humans altering their environment, especially through processes like draining aquifers, will continue to greatly change flow regime. So a brief synopsis of a very large project that is ongoing. I have many people to thank, especially Dr. Moody at Middlebury and the Moody Lab, Tina Kai, who did a lot of this research this summer, increasing this massive data set. And yeah, it's a very fun project, and I look forward to the future of what they continue to find out.
All right. Thanks, Annie. Does anybody have any questions? You can put them in the chat or just go ahead and unmute and ask them if you'd like.
Let's see if I have chat.
I don't see anything in the chat yet. Okay, I'll start with a question, Annie. This is really cool, and I'm really glad that people are looking at this stuff on the continental scale. That's really cool. Looking at your taxa there, I was wondering, are there any other metrics of food quality? Because it looks like you have some detritivores and stuff in there as well. So you wouldn't maybe imagine they would be super correlated.
You're breaking up a bit.
Sorry.
Oh, sorry. You broke up a little bit, which is, I do want to clarify, this is an image from previous research. This is not from
okay, so you're only looking at functional diversity of grazers.
Sorry, let me go. So we were looking at I assume you're talking about trophic habit. So
yeah,
we looked at if I remember back, we looked at grazers, shredders, predators, yeah, several other detritivores, if I can remember. So yes, we were looking at more than just grazers in terms of trophic habit.
And do you have either, I guess, water chemistry data or stoichiometry from the detrital pools to correlate with?
So we were using periphyton data. I don't remember if we used water chemistry data, but that was what we could gather from what Neon was collecting at those same sites.
Okay, I think Dedmer has his hand up.
Yes.
Sorry. Yeah, thanks, Annie, for great talk. And for me, it's working on lakes and more like non-moving systems. This is a bit scary stuff. And also the animals that you have there, they look quite heavy. Anyway, I was wondering, maybe you mentioned it, but sorry I missed it. But what is the relation between like drought and heavy rainfall and nutrient availabilities? Because I can imagine both directions can happen. So you have these two processes, I guess. So there's this physical stress of stuff moving away, so to say, and then we want to grow fast. But then nutrients can also maybe come in the system more easily with rainfall, or how does that work?
Yeah, I mean, from my reading, looking at the effect of flooding on nutrient availability, it's these flashes of like phosphorus availability specifically. So it favors those insects that can quickly utilize what is coming through a stream. You also see correlations between like life history traits that allow insects to quickly colonize a habitat once, especially in these intermittent streams where through droughts they dry up into disparate pools. So insects that are able to colonize streams that suddenly become connected, hydrologically connected from disparate pools. In terms of nutrient availability, I would just think like as water returns from periods of heavy rainfall or after periods of drought, it's a flash of nutrient availability. If that answers your question, I worked a little around it, but
yeah. So there are moments that you have. So it's not a direct relationship. So there are moments you expect high nutrient peaks, so to say, depending on the movement. So if there is a flooding, like, is then the retention time shortly also increased? So is the flow slowing down and so on in the system?
I mean, it's hard to generalize about because these sites are all over the country.
Yeah,
We did see
yeah.
like really interesting patterns when you look at the sites that are concentrated in the middle of the country, because you see more of that drought flood hydrology, this whiplash hydrology as it's called. But so it's hard to generalize about all of them because you see a lot of variation, which was also the point. So it's hard to, focus in on each site, especially when you're looking at 19 sites. But definitely in sites like in the Great Plains, again, like with those intermittent streams, you see, I would expect to see that those peaks in nutrient availability. Yeah, and that would obviously, quickly change the community composition of an aquatic system. Sorry, my lab mate just walked in.
No worries.
Thanks a lot.
Yeah, that was a great explanation. And Eric added that they're not currently collecting detritus stoichiometry, but since everybody knows it's like the most important thing ever in streams.
Yeah,
sorry, I'm biased. I like to try to work.
Well,
But yeah, no, it'll be
quality,
really cool
yeah.
to look at that. Yeah, so that'll be
food
really
and
fun.
food quality is so important in that conversation, and especially with all of these like detritivores insects. It's a really good point, and I would love to see what that looks like.
Cool. Well, I think in interest of time, we want to get everybody in the hour. So thanks for starting it off. You did a really great job, Annie. So I think next up we have Kat Jones, and really excited to introduce her. I guess Nicole and I can both introduce her. So she is our successor in Paul's lab and is doing really, really fabulous things. So she's been doing a lot of work on nutrigenomics. I can't even pronounce it. It's too over my head. Looking at the effects of stoichiometric imbalances on Daphnia and doing a ton of really awesome transcriptomic work. So Nicole, do you have anything to add? Sorry, I stepped on you.
No, you can go for it. Yeah, take it away, Kat. I can't wait to hear this talk and see how this project's been coming along.
Wow, okay, so thank you for that introduction. Try very hard to live up to it. Okay, so can everybody see my presentation there?
Yeah, you're good, Kat.
Yep, excellent. So my name is Kat. I'm in Paul's lab at Trent University, and I've called my talk Looking Under the Hood of Stoichiometric Imbalances. And this talk specifically is based off a paper that I'm writing for my second chapter, which we're hoping is close to being ready for submission. But this is generally the theme of my PhD research overall, which is we have these stoichiometric imbalances. How can we detect these? How do these stoichiometric imbalances affect the transcriptome? Can we use transcriptomics to actually actively diagnose stoichiometric imbalances? So just to start off, the classic stoichiometric experiment, I think that most people would probably recognize. We've got our Daphnia over here, our sort of model stoichiometric organism, relatively P-rich, and we feed it on some food, some ceston, with a higher C to P ratio than the Daphnia possesses. We measure these sort of crude physiological traits like somatic growth rate, fecundity, body carbon to phosphorus ratio. We might also look at maybe some population level traits like intrinsic rate of increase and generation time. And we can pretty readily conclude that the Daphnia are growth limited by phosphorus. This gets a little bit more complicated, however, once we step away from the lab and we're trying to apply this to wild systems. So if I was to go out to one of the many, many lakes that we have surrounding us here in Peterborough, I could definitely collect some Daphnia, I could definitely collect some ceston, and I could take some measurements of things like carbon to phosphorus ratios, but we don't have any form of baseline to detect changes to growth and reproduction. So these are two of our big indicators in lab-based experiments that we just cannot use in unknown animals. We actually don't know what they've been eating. The ceston that I sampled today might be completely different to the ceston makeup a week ago, 2 weeks ago, that these Daphnia have been growing on. So functionally, they have a completely unknown nutritional history. And finally, there might be other stressors getting involved, so other nutrients that are maybe in short supply, or even other environmental stressors like increases in temperature, which could also be having an effect on the phenotype. So my big question here is, how do we detect stoichiometric imbalances in animals with an unknown nutritional history? How can we go out and do all these awesome stoichiometric experiments we've been doing in the lab in the field and start applying some of this knowledge we've collected to wild systems? So this is kind of how I view my research. I stand by the lake and I chat with the Daphnia, and unfortunately they don't seem to be able to tell me very much because they can't talk. But when a consumer faces stoichiometric imbalances, not only are they making these coarse physiological changes like, you know, reduction in growth rate and reducing reproductive output, but there's actually a lot of nutrient specific adjustments they are making throughout the ingestion, digestion, excretion process. So things like adjusting digestion. So for example, low phosphorus algae can have tougher cell walls. So we might be looking at increasing the digestibility of our food. We might also be looking at increasing uptake efficiency. So if we're feeding on a low nitrogen food source, we might look at bumping up the efficiency with which we're uptaking nitrogen. There might be costly metabolic or other pathways that we can downregulate. So something that takes a lot of phosphorus, we might be able to downregulate and just maybe increase our phosphorus availability. And of course, we can also adjust things like excretion. So if we're looking at phosphorus limitation, we might be excreting excess carbon. So we've got all of these little internal processes that are going on at the same time as we've got these coarse physiological and population responses. So my hypothesis that I'm working on for this paper is all these changes require pathway-specific genes to be differentially expressed. Right, you can't downregulate a metabolic pathway without also downregulating the genes needed to express that pathway. Related to that, I'm hypothesizing that these changes are also specific to the type of imbalance being experienced. So if I'm nitrogen limited, primarily, if my food is not giving me enough nitrogen, I'm not necessarily going to be focusing on pathways that are going to boost my phosphorus uptake. I'm going to be focused on pathways that increase my nitrogen uptake. So although the Daphnia can't tell us what's limiting, maybe their transcriptome can. So the sort of basic experimental setup that I used was I grew Daphnia on 5 nutrient limited diets in addition to a control well-fed diet. So we used phosphorus limitation. So I think we had a C to P ratio of around 600. We did nitrogen limitation. So I think we ended up with a CN ratio in the food of around 14. We did calcium limitation. So we adjusted the growth medium that they were living in to be a low calcium environment. We reduced the amount of food we were giving them. So kind of introducing a carbon limited aspect. And then we also decided to explore limitation by more complex biomolecules. So we included a cyanobacterial diet, which cyanobacteria are generally very low in a lot of essential fatty acids. So I extracted the RNA from my animals. I also took some measures of growth and nutrient content. We sent our RNA to Genome Quebec for next generation RNA sequencing. And then I used a bioinformatics pipeline just to map my reads back onto an annotated Pulex genome and essentially count features and look at where we were seeing of regions of differential expression. We then did some further analysis to turn these global patterns of differential gene expression analysis, expression across, I believe, 33,049 genes was how many my Daphnia had in the genome. So how do we go from responses at the level of 33,000 genes down to just a few genes that we can look and tell us what's happening? So I used a variety of different variable selection techniques, looking at how we can map gene responses onto nutritional stress. And I won't go into all the boring details of that, although you are free to read the paper when it eventually comes out. But the genes that I'm going to be presenting to you today, we used an elastic net regression to select them. So first of all, before we start going under the hood, as it were, we kind of want to confirm that we have actually limited growth in our animals. So I'm going to focus on this graph on the top left here, which is mass specific growth rate per day. And the second bar here, the tallest bar, this is our well-fed control animals. And you can see to a variety of extents, growth was significantly reduced under each of our treatments. We also noticed some trends within the nutritional content, but this is a little bit less straightforward. So I'm just going to focus on growth for now and confirm all of our treatments. Yep, we definitely limited their growth. So we should get some nice results once we start drilling down into that transcriptome. So as I said, I used the elastic net regression. I ended up with 20 genes that I think could make pretty good biomarkers of nutritional stress. And I've plotted them here in a sparse partial least squares. And we can see in the middle here, the orange control sort of occupying a central position. And we see all of our treatments falling away nicely from the control. Tiny little bit of overlap here with food. But other than that, we can see these clear, distinct stoichiometric phenotypes emerging for each nutrient. So this for me was great news. This means I can take these 20 genes, I can take my unknown animal, and I should be able, just by looking at the expression of these 20 genes, be able to tell you the type of stoichiometric limitation that animal is experiencing, which to me is incredibly exciting. So I'm just going to show you this heat map now. So these are the 20 genes that my model selected. And you can see we've got some nice distinct barcodes for each type of limitation. Control animals are not on here because this is a heat map of log fold change. So this is all whether the genes were up in red or down in blue and sort of whitey color there, whether they were up or down regulated in relation to the control animals. So some of these show some really nice linkages with nutrient specific pathways. So the first gene I'm going to focus on here is the Dapidraft 301663, and this codes for the cytochrome P450, which is involved in the eicosanoid biosynthesis pathway. Eicosanoids are sort of derived from fatty acids, and they regulate things like immune response and reproduction. And in some work done a little while ago now using microarrays rather than RNA-seq, another lab group confirmed that cytochrome P450 is downregulated under low phosphorus conditions, which we can see here. We're looking at around -2 to -3 log fold change for the phosphorus. But what's just as interesting to me here is, because we've introduced these other 4 treatments, we can see that something about this cytochrome P450 that is so necessarily reduced under low phosphorus seems to be important for other nutrient limitations. Next, we've got the chitin binding peritrophin A, which, as you can see, our downregulation here is with calcium. So chitin, it's involved in forming extracellular structures, primarily in the exoskeleton and midgut matrix. Now, I was unable to do tissue-specific sequencing here because the animals are so very small, so we can only look at this on the level of the whole body. But given what we know about the relationship between calcium limitation and formulation of an exoskeleton, I think this gives us some fairly compelling evidence that we're seeing the exoskeleton being either thinner or covering a smaller surface area. And then finally, we've got these type 4 and 8 collagens. Now, collagens, they're mainly used for protein biosynthesis and, again, extracellular structures. So we don't quite know what we're doing here, but I think there's some really interesting stuff going on here with the carbon and nitrogen being downregulated. So there's something here around protein synthesis that is clearly carbon and nitrogen heavy. So why did I bother doing any of this? So I think, first of all, it really contributes to our fundamental understanding of the effects of poor nutrition on animal physiology. So not only do we know that these nutrients are essential for growth or for reproduction, we can get really specific and say, oh, well, this nutrient is necessary for the building of extracellular structures, and this nutrient is necessary for the, formation of certain enzymes. The other thing it contributes to is further functional annotation of the genome. So Daphnia is, I describe it as a newly model organism. It's not really a model organism to the level of Sienna rhabditis or Drosophila, which means that although it has been sequenced, we've still got a lot of work to do on the genome. A lot of the functions still haven't been identified. And if I go back to the heat map here, a third of these genes, so 7 of these genes, had completely unknown functions. But by looking at their expression levels in relation to different nutritional treatments, that's going to help us unpick what the function of some of these genes are. Also allows us to diagnose specific nutritional limitations based on gene expression, which for me is a really good news story when it comes to maybe in the future deploying these as nutritional biomarkers for biomonitoring programs. So I just want to thank you all for listening, and I've got some thanks to give. So firstly, I want to thank the Frost Laboratory at Trent. So Paul, my supervisor, thank you very much. And then some specific shout outs to Cody, Marcus, Olivia, Victoria and Nolan for various bits of help they've given me. Other people at Trent, Aaron Schaefer, who's an associate professor in biology and forensics, and Colleen Doyle, who works in the Wildlife Forensics Lab, both very instrumental in helping me with the molecular work. Want to thank Compute Canada. We use their SharkNet computer cluster for running the bioinformatics. And then our funding sources, so the John R. Evans Leadership Fund, auditor Aaron Schaefer, and NSERC Discovery Grants, auditor both Paul and Aaron. So I will stop there and very happily take any questions you might have.
Is there any questions? You can just raise your hand and unmute yourself. Oh, Dedmer has one.
Yeah, thanks. Great overview. Did you test it in some wild Daphnia that you picked?
Not yet, no. So we're still looking at testing robustness to other stressors. So I think we've got a little bit more work to do in the lab. But yeah, my eventual hope is that we'll be able to go out to some wild lakes and diagnose everyone with what's limiting them. Yeah, but I think we're maybe a couple of experiments down the road from that.
I can imagine you see like big impacts of like maybe temperature and like, as you say, other stressors, right?
Yeah.
Okay, thanks.
You're up, Yana, if you want to ask your question.
Oh yeah, sorry. Thanks, it was really interesting to listen to you. I got a question. So you basically show that you can identify 3 genes right now. How do you continue? What are the next steps forward to identify what those genes that are clearly distinct between the different treatments are actually responsible for? What are they regulating?
Unfortunately, not fully my area of expertise. I know to get a really sort of definite functional annotation, you have to do a lot of things like knockout studies, which is why we've only really seen this in the genetic model organisms so far, like Drosophila, where you can actually knock out genes and figure out functions that way. But I am hoping that maybe we can look at maybe co-expression networks and isoforms, and maybe that can at least help us get some cog groups. So what are the isoforms in other species and things? But I think it's a fairly long road to getting full definite functional annotations for a lot of these things.
Thank you. Yeah, that's really interesting.
Kim, you have a question?
Yeah, Kat, that was super cool. That's a really, really nice system. So I was wondering if you have any sense of how transferable these genes are to, say, other species. Like, are you like, yeah, maybe that's like all the question is. Yeah.
Yeah, so I have my own personal theories, and I also have, we've got some plans for work on testing this, certainly within other Daphnia species. My personal thought on this is that I think species might affect what limits you and to what extent, but I think the sort of the digestive architecture and the metabolic architecture is very well conserved. So we've got a little bit of work to do to deploy these on some other Daphnia species, but I'm pretty hopeful that what we're looking at is some very conserved pathways. So we were sort of talking about this recently and sort of thinking, you know, the difference between a Pulex and Magna is maybe the difference between a human and a donkey. And although we've got some very fundamental physiological differences, ultimately the way our digestive systems kind of function and the way our enzymes function is broadly conserved. So that's my theory anyway, but we do have some work planned to actually confirm that.
Awesome. Yeah, it seems like there's a lot of things you can do with this system. It's really cool.
Oh yeah.
All right, I think we might move on as it is 22. Kat, I could talk to you for days though, but yes, I'm more interested in this even now. I'm excited you got to finish off my project I really wanted to do. So it's awesome. All right, and our last speaker of the day is Patrick Thomas. He is in Germany right now, and we're going to continue on with this zooplankton. I don't know if it's going to be just Daphnia or if we're going to get some other zooplankton in this talk. If you want to share your screen, you should be able to kick Kat off.
Yep.
There we go.
Cool.
All right, so yeah,
Okay.
You can take it away.
All right. Yeah, let me know if there's anything technically not working, but yeah, hopefully it works. Yeah, so it'll be mostly Daphnia studies that are included, but still, maybe there's a few copepods in. Yeah, so this is going to be also about part of my PhD that I've been doing here at the University of Oldenburg in Germany, and it's a project that's mostly complete, but still in prep. And yeah, it's doing a meta-analysis about mineral and biochemical nutrient limitation of zooplankton. And as we all know, there's a ton of variation in phytoplankton, stoichiometric and biochemical food quality. Just throw up this quote from Sterner to summarize it, that they may be the most biochemically diverse guild in nature, phytoplankton. And yeah, there's a lot of studies I could show to kind of convey this variation, but instead I'll show all of them together. So this is the data from our meta-analysis, showing the degree of variation in C2P across just green algae in our database. And yeah, so you see that it varies really widely from below the Redfield ratio here around 100 to well above, and well above this ratio of 300, which is kind of one benchmark of phosphorus limitation. At the same time, yeah, there's really huge variation in fatty acid concentrations. So for example, yeah, EPA, one of the most important omega-3 fatty acids, can vary between 0 and a lot. So for example, in cyanobacteria, you'll have near 0 EPA. And in cryptophytes, you'll have really high EPA concentrations. And this has really broad implications for zooplankton production. So with phosphorus limitation, zooplankton have really poor growth. And with fatty acid limitation, they can also have pretty poor growth. Since we know, we know a lot about this already. What is the purpose of doing a meta-analysis? First, to get overall a quantitative summary of nutrient limitation effects on zooplankton. Second, and kind of more interesting in my perspective, is to get a look at the interactions among these major types of nutrients for zooplankton. And third, to assess how kind of the context, the environmental and experimental context can influence these limitation effects. The methods we used were just a standard literature review and systematic literature review and meta-analysis. So the criteria we used to select studies were those that had some kind of variation in phytoplankton food quality. And here you see some of what we limited the scope to, and that it only included nitrogen, phosphorus, fatty acid, or sterol variation. That the study measured some kind of response in terms of growth, reproduction, or survival response to phytoplankton food quality variation. And third, that the study controlled for really major confounding factors. For example, that food quantity wasn't really strongly confounded with food quality, for example. Yeah, and so this is the flow of how it looked like for selecting studies. We started out with over 1,300 and then excluded a lot that were not relevant or that didn't meet these three main criteria, and ended up with just about 100 studies in the final database. And we had two main effect sizes that we used to look at nutrient limitation. The first one, which we used for the more controlled manipulations, more like treatment versus control type studies, was the Hedges G, where we took the response of zooplankton in replete versus limited conditions, and it's standardized by the pooled standard deviation. So that was for about half of the studies, a little less than half. The rest didn't really have a clear treatment versus control. For example, if there's variation across, let's say, 6 species, which this data would show, where for a given, so there's across 6 species, there might be a suite of nutrients that are measured and a zooplankton response in terms of growth or reproduction or survival. And so for each combination of a nutrient and a response, you can get a correlation between those two. So it's kind of a more indirect way of looking at the data. It's kind of harder to tell what's going on, but I see it as kind of backup data to these more controlled ones where you can draw stronger inferences. So that's a quick summary of how we did it. And here's a very quick summary of some of the results. On the x-axis is the effect size for these more controlled manipulations in terms of the Hedges G, split between somatic growth and reproduction. And basically the story is that everything has strong effects. So alleviating limitation of nitrogen, phosphorus, fatty acids, and cereals all has strong, significant positive effects. Yeah, individual fatty acids have effects, as well as kind of summarizing as bulk fatty acids. Maybe there's a bit more of an effect relative, the fatty acids for reproduction relative to somatic growth. But yeah, this is kind of the broad overview. And I should have said that the numbers here represent the number of publications and the number of overall observations that we see. So you get an idea of how much data this is coming from. And in some cases, it's not too much. So the summary is, strong effects of everything. Cereals, we have a bit less studies, but also kind of strong evidence that they have strong effects on zooplankton performance. And this last set here are ones that kind of give a hint at effects of co-limitation. Again, we don't have too many studies that do this, where they have a controlled manipulation, alleviation of multiple types of nutrient limitation at the same time. So that is already one result, but so far it seems that they have stronger effects than individual nutrient interventions. So that was the data for these, the controlled subset of studies. And now here's the data for these kind of more correlative ones, which is a lot. And I don't want you to look at every single point right now, but what I take from this is that it agrees with the previous data. It's generally nice that it's backed up with these potentially, these studies where there's indirect correlation type inferences that we can make, that generally has really strong effects of nitrogen, phosphorus, and individual fatty acid limitation. We see some things like certain omega-6 fatty acids have a negative correlation, which I think could be due to the fact that these are potentially correlated, negatively correlated with omega-3 fatty acids and phosphorus, but it's a bit hard to tell with this kind of thing. So, but in general, despite everything that's could be affecting these results and be kind of confounded, it supports the simpler manipulative stuff. So that's the summary of big picture effects. There's a couple of things we can do to break it up and look at things, how different, how the context matters. And this is one that I think is the coolest and maybe more interesting for you guys, where the way that phosphorus was manipulated makes a really big difference. So for example, these studies on the top row would be those that used direct manipulation of phosphorus, where, for example, the treatment and control would both be subjected to phosphorus limitation, have a really high C/B ratio, and the replete condition would have phosphorus spiked, and then it would be taken up really quickly. And so in theory, the only thing that's changed is the phosphorus content. Everything else is the same. So any kind of morphological or physiological changes that are caused by phosphorus limitation are at least equal. So we see less effects of just phosphorus. And when you include these indirect effects as well, you have a much stronger difference between treatment and control. So I think the point is that the indirect effects of phosphorus limitation matter, and I think there's a lot to look at here that explains the difference between direct and indirect total effects. So just one quick look at how to examine these interactions between different broad nutrient types is seeing how the effect of fatty acid addition changes. So the y-axis here is the effect size, so the hedges G for these controlled studies, controlled manipulations. Yeah, and the x-axis is the CDP ratio. So in general, there's lower effects of adding fatty acids when you have really high CDP ratios. So that kind of supports what people have kind of believed and known for a while, that fatty acids become much more important when CDP, when phosphorus is replete, essentially. But also note that, most of these still have positive effects of adding fatty acids, even at really extremely high CDP ratios. So although the effect is lower, it's not zero. So I think that's interesting. Okay, and the last bit I will show you guys is comparing lab isolates versus more natural communities. And so, yeah, for this, yeah, the top row here is all lab isolates showing effects for phosphorus and fatty acid manipulation. And the point is that when you increase phosphorus or fatty acid availability to lab isolates, the effects are much stronger for both somatic growth on the left and reproduction here than they are for natural ceston. So, and also note that there's a lot less studies that have done these kind of controlled manipulations for natural communities versus lab isolates. So, yeah, I think there's also there's a ton that could be causing the differences between lab isolates and natural ceston, but I think that's just a really interesting point, that maybe there's a gap to bridge here in explaining that. All right, so that was a very quick look at some data. And what would I have as take-home message? Yeah, just to confirm that limitation by food quality, both mineral and biochemical, is very strong, rarely unidimensional. That the context can make a big difference and explain kind of the variability we see in results across studies. But I would also say that, we're probably all sick of hearing how every single thing in ecology is extremely context dependent, but I think here a little bit of context can go a long way and kind of, yeah, help make sense of things instead of kind of make things more complicated. At least that's how I hope this, yeah, will help. And finally, there's a lot more to learn from this data set, because it's already pretty big, but there's a lot more that could be done with the data that we did not use for this and data that doesn't exist yet. So, all right, with that, I would like to thank especially everyone who did all these studies, including a lot of people listening to the talk right now, and those that provided some raw data or input so far. I'd like to thank my supervisors and collaborators and everyone in our lab who has given feedback so far. And yeah, as I said, it's in prep, so any feedback or thoughts are very welcome. So thanks.
All right, great talk. Is there any questions for Patrick? Go ahead, Kim.
Hey, so thanks. Super talk. It's really nice to see you again as well. I was wondering about one of the last results you showed comparing the lab and the natural ceston. For the lab isolates, are those mixed cultures or like single species as well? Or do you know?
Yes, they're both, and I think I have maybe something else. Yeah, so it's both. I think it's a mixture of single species and mixtures, but yeah, I split it up by taxon also, and yeah, yeah, essentially, if you have lab isolates, I think I'm not sure. Yeah, I think these are all lab isolates where you have mixed communities in the lab. Even then, you have a lower effect size. So
Interesting.
I think
Okay.
diversity. I think adding more diversity is at least part of it, because then you're more likely to get something with really nice, fatty acids or something, or at least lower the effect of some Senadesmus that has like a C2P of 3,000. So.
Okay, cool. Really, really nice work you're doing.
Thanks.
Any other questions? I don't know if this is really a question or I guess just a comment, but yeah, I was wondering, I guess